working on gene therapy for decades now. And I've also been working on choroideremia because I met very important people in the choroideremia field along the way, and I'll introduce you them as I go. But some of the most important people in my life are the young scientists I work with. And here's some of the team that were involved in some of the research that you will see here today, all sporting rather retro Team CHM shirts. But the two ladies in blue are Ali and Manuela, and they did the stem cell work. Whereas Patrick was a postdoc. He's from Portugal. He's a postdoc who was funded briefly by the CRF, and he did a lot of work genetically correcting choroideremia stem cells using our vector system. And he worked with Bianca to do this. So these guys are the ones that do all the work. And as we heard earlier, I just do PowerPoint presentations and travel the world talking about it. But I think it's really important to see them. And they loved the shirts that they got. Scampered round Heidelberg proudly in them. So I also want to put this really into perspective, because this is a paper, and it's the first paper which actually described using gene therapy for the treatment of human disease. So this is only 50 years ago, and this was before we could actually do that. And we weren't able to realistically clone DNA in these days. So the choroideremia gene hadn't even been located or discovered. The molecular mechanism was still not found for 20 years after this. So this is 50 years ago this year that gene therapy was first described in the literature. And to think where we've come now, cloning genes, curing diseases in some cases with genetic elements, I think it's tremendous. And so we're really at the beginning of the breakthroughs that we're going to make in the treatment of these diseases. So my history is I left Scotland, moved to London, and I worked in St. Mary's Hospital. And this institute was a gene hunting lab. They were looking for the genes that cause disease. And around the same sort of time as the gene for choroideremia was discovered, they were looking for cystic fibrosis. And although the gene was found just across the lake in Toronto, it was in the fridge and freezer in this hospital. And as soon as they found the gene, they wanted to do gene therapy for this. So I started work on gene therapy for cystic fibrosis. And this is where my life and my gene therapy life collided with choroideremia, because my first head of department moved to Australia, and my second head of department was Professor Miguel Seabra, who moved to this institute and took over our department. And it was he, a little later on in our careers, that he kind of adopted my research team into his own, and I was working alongside him and Tanya working on choroideremia. So in those days, I was interested in delivering DNA to cells, so mainly for cystic fibrosis into the airway epithelia of cells. And we got really good at delivering DNA. And so here's a little baby mouse, and we injected it with a gene which can express luciferase, so it makes the mouse glow. And we can measure the emission of light. And these were the best DNA vectors we could use, and we could deliver them really nicely. So you can see the little mouse on the left is glowing. It's got gene expression everywhere. But after no more than 1 week, it stops glowing. The DNA disappears, the DNA is silenced, the cells which had that DNA are damaged or lost. So I realized at that stage that I really wanted to work on the DNA. I didn't want to work on the formulations. I really wanted to improve the DNA so that we could deliver genes, we could cure cells without causing them damage and without having to do it over and over again. And so that was the turning point after this work. Turns out, Annette, I was recruited from London and moved to Heidelberg. So this is little Heidelberg. Many of you might have been there. You should definitely go if you haven't. It's a beautiful town. And I was recruited by this man. And some of you might know him, but you probably won't know him. His name is Harold Zurhausen. And he probably single-handedly has prevented more people in the world getting cancer than any other. So he got a Nobel Prize for discovering that human papillomaviruses can cause cancer. And because of that discovery, he developed a vaccine for it. So he was not only just discovered the cause of cancer, but also a preventative measure. So I think this was pretty exciting when he recruited me. I didn't really understand why he wanted to invite the Nonviral guy over to his department, but we had some discussions about it. But the main thing is when he took me there, his whole department was filled with people who try and understand the mechanisms of how pathogens can damage cells. So if your cell is infected with a virus, the cell knows about it. There can be damage on the outside if there's antibody responses. and there could be damage on the inside of the cells because of innate immunity. So being infected by a pathogen is not good for the body, it's not good for a cell. And his whole department, Harold's whole department, was working out how viruses cause cellular damage. So I want to talk a little bit about the virus in the room. And sadly, almost certainly, this virus is going to be in the room or nearby. And this really is taking it home to us. This is the COVID, SARS-CoV-2 virus. This really illustrates to us how little we understand of viruses and how much damage a simple virus can cause us. And this virus has, of course, caused havoc in our lives in the last 2 years. And we still don't really understand why it causes so much damage. But it's an infectious agent of humans. It causes all sorts of disease and damage. And, we're still struggling to understand that. The real virus or elephant in the room is the other one, and it's adeno-associated virus. And it's not that I'm railing against this, because this is an amazing genetic tool, and it's been at the forefront of genetic research and gene therapy research for a decade now, and it's transformed many people's lives. But it's also an infectious agent of humans. It's the reason why you can use it once and then you have an immune response against it. It's also the reason that it can cause inflammation of cells where it's delivered to. So, people who've been delivered, AAVs being delivered to their eyes, they had anti-inflammatories before delivery, they had anti-inflammatories after delivery. And it's all down to this as an infectious agent, and your cells know about that. And because of that, there can be repercussions and there can be damage to the cells. So I'm not saying that, it's not that this was a bad idea to use it, because it absolutely works in some systems. It might just not be the best system or vector choice for choroideremia. And I think it's amazing where we've come. We can apply these, and we've been able to develop such tools. But we need to think about alternatives. We need to think about different ways to deliver DNA to cells. So we all know there's a variety of approaches. We have viral vectors, and I won't go into so many details, but there's a range of viruses that have been modified for gene delivery. And there's also non-viral vectors. And then I said I was naive before I went to Heidelberg about how safe these were, because we actually make these in bacteria, so they're also pathogenic DNA. They can cause damage to the cells themselves. And so we've worked tirelessly in the last years to make these vectors less damaging. So there's a range of them. You might have heard of transposons, where they integrate DNA into the cells. There's EBNA-based vectors, and we use these ones called SMAR vectors. And we're using these to develop an alternative platform that perhaps will be safer. And our vectors live outside the chromosomes. They don't integrate. So our vectors, in principle, don't cause any cellular damage. They don't cause molecular damage or genetic damage. And so that's the important thing. So this is it. So I'll start talking about these vectors. So these are simple, non-viral, non-pathogenic DNA molecules. They're really simple to manufacture. They're efficient to deliver, and they remain outside the chromosome of the cell. They have unlimited capacity. So that means we can do quite sophisticated genetic modification to them. We can put huge genes in, because choroideremia has got a small gene, but still we only use the cDNA. We don't use the full genomic sequence, we use the cDNA. And in principle, we could really use the whole genomic sequence. We could use the whole genomic promoter. We could allow the cell to control the expression. And we can also add some extra elements if we wish. We can use these vectors in vivo, in vitro, ex vivo. So in vivo is directly in an animal or a patient. We can use them in vitro, so that's in culture in the cells. Or we can modify cells in culture and then put them back into an animal or a patient, and that's ex vivo. And they have mitotic stability. So that means they replicate with a cell. So stem cells are rapidly dividing. As we heard earlier, they can divide forever. And it's really nice that our vectors can go with them. So we can modify one single cell and it divides for the lifetime of that cell and its population. And I'll show you some work with that. And the other nice thing is the DNA is not silenced. It's not interfered with by epigenetic effects. So the expression should last for the lifetime of that cell, which is really useful. The other thing, as I mentioned before, is not immunogenic. So in principle, if it does ever switch off or we want to treat some new cells, then we can repeat administer. So we could do some additional administrations. So as I said, there's a range of application, and these go from cell delivery, gene therapy, and a whole range of applications we can think of. So as I said, there are alternatives. Why use these? This is just another illustration. On the top, we had some normal human cells. We introduced a DNA to make them green so we could see them. 2 weeks later, the green had disappeared. with a normal vector. So the vectors I took from London were like that. The new vectors, this is after 6 weeks. The cells are rapidly dividing and they're still green. So this works in culture for standard cells. So we use these molecules on our vectors called SMARs, and that allows the DNA to stick into the nucleus, not integrate into it, but stick to it, and it allows it to multiply and replicate with the cells. I mean, these slides will be available afterwards if you want more detail, you can ask me. So I don't need to go into all of these details, but this is the important element. There are these SMARs, which allow the DNA to function as we want. And we've done this. This is, the end of my work in London. If we inject it into mouse, this is once again expressing light. So on the left-hand side is a mouse expressing light from its liver. And on the other side is actually, we did subretinal injections using this more or less the same construct or same expression cassette that's in an AAV. And we injected into the right-hand mouse, the left-hand eye, and it's also expressing light. And it turns out that this expresses for the lifetime of these animals. So one single injection, lifetime expression. I think this might be a bit small to see, but this is day 365, and these mice are still expressing in their eyes. So this was what we were hoping to do for choroideremia. Circumstances, funding, the rest of it meant that we didn't actually do this, but others did. Some of our collaborators have successfully managed to treat genetic diseases in mice in the eye using vector systems like I'm describing. So these are 3 papers. Stargardt's and Lieber's congenital amaurosis, and just a nice comparison between AAV and these vectors. So these have been used by other groups, not just us. It's not just me standing here saying these are the best vectors ever. Other people use them too. So I want to give you some examples. And as I said, that Maria Musagi will be talking afterwards. And we've split this talk because she's the ophthalmic surgeon. She's the famous ophthalmologist who's actually using our vectors for choroideremia. So I'm going to tell you about the vectors, and I'm telling you maybe about some of the things that I do in my lab, and they will infer that they can be used for eye disease. And I actually show you some nice choroideremia work, but most of the choroideremia discussion will be done by Maria afterwards. So what we did is we took these vectors and we thoroughly overhauled them, because with an AAV or a virus, you can't stop it being a virus. It will always be an infectious agent that the cell can recognize. But with DNA, you can change the sequences so it doesn't damage cells. And so that's what we've done. We've refined every component of the vectors. This is kind of detailed, but we don't use antibiotic resistance. We use a interfering RNA that allows our DNA to grow in the presence of sugar or absence of sugar. So we're using sugar as a selection rather than antibiotic gene. And for clinical application, you don't want to introduce antibiotic genes into patients. And we can produce this DNA to GMP standard in our lab. So that's one of the reasons I'm showing you some of the applications that we're using, is because we're going to be doing clinical trials later this year. And so our vectors will be in patients and we'll be able to demonstrate their safety. So what can we do? So we heard a lot about stem cells, and I want to show you how our vectors can be used about stem cells. We talked about stem cells earlier, but we didn't talk about genetic modification of them, because that's important. You need to be able to transfect or genetically modify a cell, turns into a stem cell, and you might want to turn this into a differentiated cell or an organoid or a new tissue. And maybe if you want to do gene correction, you want that gene to travel with the cell on its journey. So we can do this. We can take standard mouse embryonic fibroblasts or human ones, and we can differentiate them, make them green, and we get green differentiated cells. So I'll tell you about this. You can also do this with embryonic stem cells, human and mouse. This is a little twist. We also use skin, so fibroblasts from skin, but we've working with some patients and younger patients, children, infants, that, it's quite tough having a bit skin patch removed and fibroblasts removed. It's not nice. So we can actually prepare cells from urine. And these cells we can turn, there's actually stem cells in there, but we can actually take some cells from urine, we can reprogram them. And we've done this, and we can turn them into iPS and then differentiate them. And they work identically to the ones that you might get from a skin cell. So they're very useful. So what can we do with them? So the main point is I want to take a stem cell and I want to genetically modify it. This panel on the right-hand side is really a progression of vectors. The first one on the left is the original vector I was using in London. You can see this is time down here. So it's stem cells genetically modified with a range of vectors. the original vector here, this one, it barely turns the cells green, and by day 90, they're more or less gone, or they were never there in the first place. But the last two, the smarter, this P-Smart and the nano-smart, these nanovectors, they're really green to start with, and we can turn them, many days afterwards, they're still green, and they will always be green. They will just stay green forever. No selection, no forcing the cells to be green. They're just green, and the DNA is still episomal. It's not integrated. It's not caused any damage. And so what can we do with this? So we can analyze them. And with this, you heard about pluripotency earlier, where you want to measure whether your cells can actually have the capacity to turn into other cells. All of our cells are completely identical to untreated ones. They're fully pluripotent. And this graph on the end here shows we can push them into hematopoietic stem cells in culture, and they still remain green. But the biggest test is what can we do with them? I mean, are these cells damaged by our vector? Are they changed? Are they behaving like a normal stem cell would? So yes, they are. So we take embryonic stem cells, and this is from a beige mouse, and we can make them green, and we can inject them into the fertilized egg of a black mouse and then retransplant those. And it turns out we get these transgenic mice that are green. So the stem cells that we introduced at the egg have turned into an adult mouse. And so for the lifetime of the mouse, they stay green. So imagine this was a choroideremia gene or another gene. We could do this from a single cell all the way through to an adult mouse for the lifetime of that mouse. And the transgenes they're expressing, these cells are not damaged. There's no immune responses. There's no inflammation. There's no hydrocortisone, any of that. These cells are genetically modified forever. And when we look at these tissues, the DNA is still episomal. There's still circles of DNA we can purify. So this demonstrates that we can, the things we heard about earlier with stem cells, we can genetically modify them using our vectors. They should make nice retinal cells that would be implantable without any risk. The other thing you can do is pronuclear injection, and this shows the activity of it. And this is Ali here, injecting our DNA directly into the egg. So we went through a cell first and injected cells into an egg. This time we're injecting DNA into an egg. So this is what we saw yesterday with the transgenic pig. If we take these cells, we inject GFP vectors, we can actually culture these egg cells in culture, in plastic, and they will hatch. And unfortunately, they can't do much more than hatch. They won't progress more than that. But you can see over time, these cells are dividing every day. And at the end of this, thousands of cells, they're all green. And it's clear that we haven't damaged the cell. These cells are progressing, multiplying, and the DNA is going with them. But if we transplant these eggs into a mouse, we get green tissues as well. So once again, from direct injection, our vectors are contributing to the mouse, the adult mouse, and all its tissues. We can also do this with human cells. So it's very important to do it in a mouse model, but it works almost identically with human cells. So the iPS cells I showed you earlier, we can do the same. And this is pluripotent markers are all intact. We can differentiate them into hematopoietic cells, and they all remain transgenic and pluripotent. So just so Maria's not going to talk about this, but we're doing this with her, and we would like to do this. I mean, of course, she's going to do it better. But as we heard earlier, we can take choroideremia fibroblasts. So this is some skin cells from a choroideremia patient. So there's no REP1 in these cells, it's mutated. We can make iPSCs, so pluripotent stem cells from this patient. This is a great resource because these cells will divide forever. They're really a cell line. And using our vector system now, we can introduce REP1 back into these choroideremia cells. And we did it just to make it easier to see. We had REP1 and green. So we can make choroideremia cells corrected with REP1 and we make them green. Well, unfortunately, when you take black and white photos, it's white. But this shows over a whole range of days. We transfected some cells, left them in culture, and we can see, as you saw before, they form these rafts of cells. And the white expression is actually the fluorescent marker going. So there's a long time expression in cells of green. So they're also expressing REP1, as we'll see later. If we actually do FACS analysis of it, you can see, so this is after 70 or 80 days. Can't see, the screen is so small. But you can see FACS analysis actually measures single cells and shows if there's any expression. We have over 97 or 90 something % of these cells are still expressing the transgene from right back at the beginning. And more importantly, this first lane of this Western blot is choroideremia cells with no REP1. The third lane here is the choroideremia cells that now have REP1. So we put REP1 back into this. It's been for 90 days in culture. These cells are growing quite happily. They're molecularly and phenotypically identical to the original cells. Apart from now, they have REP1 and the GFP. They're undamaged, non-inflamed, and they will express REP1 forever. So what I really want to do now is take some of these cells and give them to some clever people that can turn them into retinal organoids and see if we've got genetically corrected ones. So that's the intention. So we know we can do organoids. I collaborate with a group in the Netherlands and the Erasmus Institute. So this is more or less identical, but now these cells are green. They have a green microscope, a color microscope. So this is day 67. These are iPS cells, which are green. And these guys have turned them into kidney organoids. So they're very interested in all the structures of a kidney. And so they form kidney organoids in culture, more or less like we've seen, we can form little eyes or eye organoids. And as you can see, after 87 days after we introduce the DNA, millions of cell divisions, we now have a kidney organoid that's expressing a transgene. So this is a proof of principle that this will work with the RPE and photoreceptors in the eye. So these are just two papers that people use our vectors now, because when you modify cells with our vectors, you can put them back into things with immune systems. So these are really high impact, really high level research groups have been using our cells because they know that they can use them for immunological studies afterwards. And so this is just an example that where our vectors can be used. So this platform can be used in a range of things, gene therapy, cell therapy, and these guys, if you're interested, you should look at them, that they can use our cells because they're very, very useful. The other thing, and this is just the last minute, but this is what most of my work is involved. So I work in a cancer research in Heidelberg, a cancer research institute in Heidelberg. And this is just a proof of concept. Well, there's more than a proof of concept because this is going to go into the clinic, but this will show you that we can modify cells that can be clinically useful. So in oncology, one of the most promising treatments for a range of cancers is immunotherapy, where you take a patient's immune cells out of you purify them from the patient's blood, and you can retarget them against a patient's tumor. And then you can put the cells back in to target the tumor. So we're using our vector system to do this. And this is what we've got clinical approval to do. So the regulators think our vectors are safe and useful. We can modify cells. And so I'll just whiz through this. But so you can add a receptor to the surface of the cell using our vector. And when we do this, we can do 5 times 10 to the 8, so billions of T cells purified from a patient. We can target them using our vectors. We get over 90% positive, and there's no inflammation and no damage with them. And the main thing is that we can do this with clinical approval. And when we do it, our cells can target and kill tumor cells very effectively. And the important thing, and this is really what I wanted to show you, they do this without changing any of the molecular behavior of the cell. So normal genes in a cell are not changed. So the cells are almost undamaged because of this treatment. Whereas if you use a virus, which is the bar on the right, lots and lots of genes are modified. The cells, T cells, know when they're infected. They're immune cells. These cells are, when you infect them with a lentivirus, then they're very aware of it and their genes are dysregulated and they're less happy. And because they're less happy, they're less effective. We need 5 times less cell to kill a tumor than using a virus because the cells are more effective and more functional. And this works in mice. We can kill tumors in mice more effectively too. So we've got a whole GMP manufacturing protocol. It takes us a couple of weeks to make DNA and target things. So I just want to demonstrate that it takes a long time to purify and to develop a gene therapy vector. But we already have this in place using our vector system and cell manufacturing, which will go through the clinics. And I think that will be a stepping board that we can then use our vectors for other things. So finally, over with that, the take-home message that I think these vectors are a real alternative. They combine mitotic stability, so that means they divide with the cells with really low immunogenicity, that we can produce them really at high yield, at high quantity, high quality in a lab at GMP quality. And they provide robust transgene expression in every cell we've tried and every model system we've used. So now, and it's taken a long time, maybe 20 years of my my career, but we've got efficiencies comparable to state-of-the-art clinically used viruses. And if you want long-term expression, then these will certainly work comparably to AAV and adenovirus and lentiviruses now. And so this is what we're hoping to do, is get these into the clinic and share them with researchers here. I mean, I don't work in an ophthalmological lab anymore, but I am a vectorologist, and I'm happy to share it with the people who are using AAV. They can now use vectors like ours, especially in stem cells or in gene therapy vectors. So I have a range of collaborators, thank you, but primarily my friends in the field are Ian McDonald and Maria Musiji. They're big supporters. They have our vectors. They've been using them for years, and I'm thrilled that they do. And they're really the gateway to meeting this wonderful foundation. So thank you very much for your time. And I'm happy to take some questions now. If not, we can go straight over to Maria's talk, and then we can maybe have some questions at the end. It's up to the organizers, though.
Hi, I was wondering if there was a book you would recommend to help us understand this better.
Well, there's some papers.
Because I read a book on gene therapy.
Yeah, I do have one. Yeah, there are some, and we've
really
actually contributed to one that might help. It's not too highbrow. So yeah, I would be happy to.
Okay,
I'll dig something up for you, and I can post that. I can hear you. Yeah, okay, absolutely.
There's another question.
On what you can express from these replicating vectors. So you use POL2 to express proteins. Have you done POL3 for like siRNAs?
Yeah.
And also what I guess there's a couple questions here, but payload. So how big of a gene can you put into these? And then how many copies do you get per cell?
So we can express everything and anything. There's multi-domains in this. So we've got these insulator elements that we use now to separate the domains in the vector. But as part of the vector development, we're working on making the message also very stable and non-toxic. So we have clever splicing that can actually produce silencing RNAs. But it's a little complicated from one vector to be in two transcription factories at the same time. We've found that it can disrupt the vector. So that's what you need if you want to express two completely different molecules from one vector. But we can do it, and they do seem to shift around. So that's the first question. The second thing is we have put the entire LDLR locus into it. So it's around 150 kb. We injected it into mice, and it functioned identically to the genomic sequence. So it's really a matter of handling bits of DNA like that. So we made a bacterial artificial chromosome that was, certainly over 100. Yeah, it was around 150 kb. So now, even though I call these nanovectors, they are nano because the bacterial sequence is that they're minimal or none. We can have a huge cargo. So in some of these things, we were talking about we put a CAR or TCR, but we also put tumor microenvironment influencing sequences. We can go up to 20 or 30 kb easily for some of our immunotherapy vectors. And we put the cDNA for ushers in there, and that's 16 kb. And it works nice in these cells. It works. So there's no real restriction, just our capability to handle DNA. So a physical handling thing that genetically they will work. And what was the last thing?
How many copies you have per cell?
So that establishes itself because the transfection efficiency isn't high. You might hit the cell with 1,000 copies of DNA. In the end, in these stem cells, they're around 5 to 6 copies. That's the stable number. And all of them are active because the way it works is they bind to transcription factories. So that's where replication happens too. So that's where they live. They stay switched on and they live there. But no more than half a dozen, I think. Any other questions? Okay, so are you going to introduce Maria? Okay, and then we can press the button. And Maria, as if by magic, will appear.
Hello again. Dr. Maria Musiji joins us remotely from University College London, where she holds the position of consultant ophthalmologic surgeon and clinical academic ophthalmologist. Over the past 10 years, Maria's research has focused on investigating the molecular ideology and disease mechanisms causing congenital eye anomalies and inherited retinal disease. Dr. Musiji has attended several of our conferences and continues to champion choroideremia research. Thank you. Thank you.
Hello, everyone. Thank you so much to the Choroideremia Research Foundation for inviting me to speak today. I am so sorry I could not be there in person to meet the choroideremia family, but hopefully next year. I want to thank Richard Harbottle for his brilliant talk and for giving you a real overview on the technology and potential behind DNA nanovectors, which may be the next generation of gene therapy vectors to emerge for inherited retinal diseases. I want to follow Richard by highlighting the results of our application of these DNA nanovectors in choroideremia disease model systems in our lab. But before I do that, I just want to spend a few moments looking at the bigger picture. So I'm just going to share my slides. Good, hopefully you can all see that. So inherited retinal diseases are the leading cause of certifiable blindness amongst working age adults in the UK and in the Western world. We know that there are over 360 causal genes, and choroideremia and the CHM gene being one of them. And these genes, when they're not working properly, lead to progressive sight loss and can contribute to over 80 syndromic forms, and still there are very few treatments available. We know that we have the first approved retinal gene therapy, voretigene neparvovec, and this is for patients who have molecularly confirmed biallelic RP65 changes, and it uses an adeno-associated virus subtype 2 to deliver the healthy copy of the gene to the retina. And there are several other gene therapy trials underway. The safety profile of the AAV2 vector is relatively well known, and we do know that it has a good transduction efficiency. But I wanted to talk to you about my real world experience, and in particular, a child called Leo, who was referred to me at 18 months of age. His parents reported that he wasn't looking at their faces from the age of around 4 months. He wasn't tracking objects. He couldn't see small toys. He started walking around the age of 12 to 13 months. He couldn't see very well in dim light, let alone in the dark, and he couldn't discriminate between yellow and red toys, and he preferred staring at lights. He had genetic testing, and he was confirmed to have 2 pathogenic variants in the RP65 gene. So he went on to have the gene therapy at the age of 3 years. As you know, you have to give pre, peri, and post-operative steroids to the patient. They undergo a vitrectomy, macular detachment with a subretinal injection under intraoperative guided OCT to inject the vector subretinally. And you can see that very clearly in this video by that lovely bleb. And the results of this patient, well, at 6 months, his vision had improved from 0.9 logmar, where the red arrow is showing, to 0.5 logmar, which was incredible. And he could certainly see much better in dim light, and his navigational vision was much better. But by 18 months post-op, his parents started to report that they felt that his vision and his visual behavior had deteriorated more. And indeed, when we took the measures, his vision was now 0.6 logmar, not much of a change, but then there may have been an element of better compliance. Now he's older and we're able to take better visions. But interestingly, he started to develop bilateral outer retinal atrophy. And this is thought to be related to the gene therapy itself and drug-induced. And interestingly, at ARVO this year, Dominic Fisher presented the initial results from the PERCEIVE study, which is the real world outcomes of voretigene neparvovec. And he reported several cases where they'd seen this drug-induced chorioretinal atrophy. So there are still things that we don't know about AAV vectors, things that are emerging from our experience of delivering these vectors to patients. A lot to learn. We know that there are other limitations, so that AAV vectors have a gene size limit, a carrying capacity of less than 4.7 kb. They are associated with immune inflammatory reactions in 10 to 20% of patients, which can lead to severe sight loss. And there have been variable results in trials with loss of efficacy in the choroideremia trials. And so we really do need to develop alternative approaches. Now, Richard would have very eloquently told you about the benefits of non-viral gene therapy, but just to reiterate, we are using DNA plasmids, and in particular nanovectors, which have unlimited cloning capacity, which means we can hold genes of any size. It has very limited toxic, if no viral components, and they hold a special component called a scaffold matrix attachment region. And when you put this motif, an AT-rich motif, into these vectors, it allows these vectors to have episomal maintenance, it prevents epigenetic silencing, and confers mitotic stability. So it's already been used in the retina, and this is some of the work that has been published, but these SMAR DNA vectors have been used to deliver the RPE65 gene to RPE65 knockout mice through subretinal injection. And it's shown expression of the human RPE65 protein at up to 15 months in the retinal pigment epithelium, with improvement in cone ERG measures. And approximately 20% of the RPE cells expressed the transduced gene, and there was not really any difference between transfecting with naked DNA vectors or the use of nanoparticles. But they have found that photoreceptor transfection is more challenging. So we are obviously focused on choroideremia. And as you know, this is an X-linked chorioretinal dystrophy caused by mutations in the CHM gene. And this gene encodes a protein called Rab escort protein 1, which has a cDNA length of 1.9 kb. So it's a small gene. REP1 is found in every cell of the body, and it's involved in intracellular trafficking and lipid prenylation of Rab proteins. And unfortunately, a recent phase 3 choroideremia gene therapy trial did not meet its endpoints, although we do know that further investigations are going on to see if there was actually some efficacy, but not meeting FDA standards. So in our work, we're using two model systems, the zebrafish, and you can see here a cross-section of a normal healthy zebrafish at 5 days post-fertilization with a nice lens, a very nice laminated retina, the optic nerve, and then a choroideremia zebrafish eye, which shows at 5 days widespread degeneration, loss of retinal lamination, small cataractous lenses, lots of pycnotic nuclei in areas of RP, hypertrophy, and atrophy. And this model is caused by a nonsense mutation, and there is only a single rep isoform, hence this widespread degeneration that not just affects the eye, but the rest of the body, and leads it to be embryonic lethal by day 5-6. And then the other model system we used are choroideremia patient dermal fibroblasts. So these are from skin biopsies, again from a patient with a nonsense mutation. But as I mentioned, as REP1 is expressed in all cells, we can nicely use these cells to test therapies quite easily in the lab setting. So stage 1 was obviously to develop our SMAR DNA nanovector containing the CHM cDNA, which encodes for REP1. And we did this using in-fusion cloning. And Richard would have gone through this in a lot more detail. But we created several prototypes, mainly with different promoters. So some with ubiquitous promoters and some with retinal specific promoters. And We used these initially in HEC293 cells to validate their efficiency and see which ones seem to be better. And the overall result was by far the CAG promoter showed the highest transfection efficiency in the cells that we used. And you can see that highlighted by the Western blot and the graph with the markings of VS1, which was the vector with the CAG promoter. So we then took those CAG promoter DNA SMAR vectors, and we transfected our choroideremia patient fibroblasts using electroporation with the neon transfection system. And we then looked at those cells. At 48 and 96 hours post-transfection, we had quite a large number of fibroblasts expressing GFP. And in our vectors, we have a reporter gene expressing the green fluorescent protein so that we can actually see the cells which are not only expressing GFP, but also REP1. So we then FACS sorted these cells and expanded them further. And then we conducted Western blots on those cells at day 11 and day 35 after FACS sorting. And we detected REP1 in those samples, which were not detectable in untransfected cells. So we know that the plasmid vectors are working successfully in these cells. We then wanted to also test to see if the REP1 that was being produced actually had good function. So we undertook prenylation assays, and this is where we try to assess the amount of unprenylated RABs versus prenylated RABs in our transfected versus untransfected cells. And actually what we found was a 75% improvement in the levels of prenylated RABs once we had transfected our cells with our DNA nanovectors. So really good rescue in protein function. So then we moved on to our choroideremia zebrafish. Now, I mentioned that this is a well-studied model system in our lab. You can see by this whole mount image that the whole of the zebrafish is affected by the loss of REP1. It develops a small microphthalmic eye with a widespread retinal degeneration. The body is affected with a curved spine, pericardial edema, abdominal edema, persistent yolk sac, and an uninflated swim bladder. And as I said earlier, it's embryonic lethal. So what we did is we injected our SMAR plasmid vectors into our zebrafish at the single cell stage. And then we monitored our fish until they were day 7, 8 to see whether or not the transfection of these SMAR REP1 plasmids made any difference to the phenotype and the survival of these fish. And so what we found was that after 24 hours after transfection, we immediately began to see zebrafish embryos that were expressing GFP protein. And then when we followed these fish up to day 5, we can see widespread GFP expression throughout the fish, albeit patchy, and that's because of the mosaic uptake of the nanovectors, but nonetheless widespread transfection throughout the transfected fish compared to those that were untransfected. And again, interestingly, we found that the CMV promoter was more efficient in the zebrafish, whereas remember, it was the CAG promoter which was better in the cells. So when we did cryosections and we stained for GFP, we saw very nicely GFP expression throughout the photoreceptors in both wild type that had been transfected with the nanovectors, but also the choroideremia zebrafish. And then when we undertook Western blot using the J905 antibody that is more specific to zebrafish REP1, we did not detect any REP1 in our, just generally in our choroideremia mutants, we do see a nice band in our wild type, but when we use a human-specific antibody, we do not detect REP1 in wild type or choroideremia zebrafish, but we detect REP1 in our injected choroideremia zebrafish, which has the human REP1 protein being expressed. So this is really encouraging at day 6. And when we looked at survival, we see a statistically significant increase in survival in our zebrafish that were treated and injected with the SMR vectors compared to those uninjected fish. Again, if we look at the retina, we can see nice GFP expression in our injected fish mutants. If we look at other markers such as rhodopsin and PNA for cone photoreceptors, we can see in the untreated fish, it's rather mislocalized and patchy, but when we've treated the embryos with the SMR vectors, we can definitely see restoration of a more normal pattern of PNA and rhodopsin staining, and we see better lamination of these retinas. So what's our future work? Well, we now have to really think about whether we can translate this to patients and into the human retina. And one way of doing this is thinking about various different nanoparticles that might increase the transfection efficiency of these SMR vectors into photoreceptors and RPE and even the choroid in our patients. So one of the things that is underway in the lab at the moment is growing choroideremia patient-induced pluripotent stem cells derived retinal pigment epithelium, and we're going to be transfecting this RPE with our choroideremia vectors, both with electroporation and with various nanoparticles to see what's safe and efficient. In the past, we have done some preliminary experiments using wild-type mice where we undertook a subretinal injection, a single one, and followed the mice to look for luciferase as a reporter gene expression in the retina, which we detected up to a year in these mice. But when we did this, we coupled our SMR vectors with jet PEI. And unfortunately, when we examined the back of the eyes of these mice, it appeared very toxic, and you could see these precipitates which were forming white plaques in their retina. So this is why we really do need to find something safer and more effective. And so what we're also doing is rederiving the choroideremia mouse at the Francis Crick. It's happening as we speak. So we have the choroideremia flox PGK Cree line, and we're going to be undertaking subretinal injections with our nanovectors, both neat and coupled with nanoparticles, to see if we can improve that transfection efficiency and do some longitudinal experiments. So to conclude, we've shown proof of concept that these DNA nanovectors may be a viable gene therapy for choroideremia in the future. It certainly shows rescue of the disease phenotype in patient fibroblasts and in the choroideremia zebrafish. We need to think about applying this to other disease models for choroideremia. So we're going to be moving into iPSC, RP, and even retinal organoids and the choroideremia mouse model. And we hope to we hope that this may offer a safer and more effective method of gene replacement that permits repeated delivery due to the lack of viral components, and in the hope that it may be safer for the retina in the future. And if it is, then on a wider level, it may be applicable to other inherited retinal disorders without any limitations on gene size. And as I mentioned at the beginning, could be the next generation in vector delivery for the future. So on that note, I would really love to thank my team. Dr. Maria Toms and Lies Tualbi, who have done the majority of the work that I presented today, but also thanks to Cecile Medjicase, Dr. Hadra Sarkar, Dr. Nick Owen and Katie Linkins. And then a big thank you to Dr. Richard Harbottle for all his support in developing this vector and for the continued work in getting this to hopefully be a viable treatment for choroideremia in the future. And a big thank you to all our sponsors, especially the Choroideremia Research Foundation, the Choroideremia Canada Research Group, and France Choroideremia. Thank you so much.